named professor of medicine at NYU. He's also the head of the cardiology division there in Manhattan, beautiful, overlooking the East River. And has done some fantastic work in terms of his history and credentials. I got an MD from Stanford, and then Mass General, I remember, for residency. And then I think he was in all in New York, Columbia and Einstein. Finally found his home at NYU. And he's done some really beautiful work and seminal work on conduction in the heart and arrhythmias, and most notable on the conduction system of the heart. And we have exchanged a lot of ideas over the year about what controls conduction, and you probably will hear more from him.
So
Thanks for coming.
Well, thank you. It's I used to enjoy being introduced as the new assistant professor who was on a roll. It's disconcerting, Agent, but so it is. Anyway, it's really fun to be here, seeing Yoram and of course Stacy, who I'll tease a little bit later on in the talk, and meeting old friends and meeting some new people. I've had a really fun day today. So I'm just going to give a slide or two to give a little clinical context to what our interests have been in the laboratory. Arrhythmogenesis is really complicated, and there are many ways to get there. I think in some ways some of the easier models to understand are primary channelopathies. You have a mutation in an ion channel or something regulating an ion channel, and at least you know what the proximate cause is, even if figuring out the molecular pathophysiology is more complicated. And there are many examples of these sorts of heart diseases, the channelopathies, hypertrophic cardiomyopathy, dilated, where we may have single gene defects that lead to the phenotype. And then there's ischemic heart disease, typically coronary artery disease. I joke when I made this slide, this looked like some old guy, and he's looking more like my age, which again is disconcerting. But these complex, common diseases are much more difficult to unravel. And we've been interested in both of these pathways. The problem in our field has been that pharmacotherapy to diminish the risk of sudden cardiac death and arrhythmias has been miserable. This is just one example. I'm sure other speakers have shown this over the years. The SWORD trial, where compared sotalol to placebo, and for the careful observer, you'll see that the placebo was superior to the drug treatment here, which is not an ideal approach for drug therapy. And although our clinical electrophysiologists, and probably some of yours in the hospitals, are all very happy about the implantation of defibrillators because these are big ticket items, this has kind of become the default therapy in addition to ablation for people at risk of sudden cardiac death. There are obviously some clinical problems with that. I list a few of these here. But for the scientist, it's more sort of a defeatist attitude that we can't really get a handle on molecular pathogenesis and come up with a silver bullet for it. And I suspect we may never, but many of us in this room are still trying to do that. So we have been especially interested in the Purkinje fiber network. This is a really lovely picture from 1906 by Tawara, looking at the human heart and trying to pictorialize what the Purkinje fiber network looked like here. And it's really interesting that these Purkinje fibers over the past number of years have been implicated in a fairly broad range of arrhythmogenic syndromes. Some of these are inherited syndromes like long QT, CPVT, and familial VF with mutations in transcription factors. But there's also increasing evidence that abnormalities in Purkinje excitability may trigger arrhythmias in some of these common acquired diseases, in congestive heart failure, drug-induced long QT, and ischemic heart disease. And from some of my conversations today, I know a number of you are working in these areas. This is a picture from a paper we published in 2014 with a reporter gene, a green fluorescent reporter gene in the mouse Purkinje fiber network. And I think it just tells you that the more we learn, the more we just recapitulate history. So here we have among the most sophisticated techniques you could think of, and it doesn't look all that different than looking under the microscope and just drawing it. So there are a lot of lessons to be learned from history, and I think I learned that more and more over time. So just some examples about Purkinje cells as arrhythmogenic triggers, and we've worked with a number of mouse models over the years. These are calcium transients in ventricular myocytes. These are genetically engineered mice that have a CBAC mutation or ianidine receptor gain of function mutation. And you can see here that the Purkinje cells develop these after depolarizations. And this is a long QT3 mutant mouse model, and these develop early after depolarization. So we can recapitulate a lot of what's seen in human diseases in some of these mouse models, and this has spurred some of our work forward.
I have a question. I'm going to interrupt you. I apologize. Because I don't do this for a living. If I look at a Purkinje cell under a microscope next to a myocyte, will it move?
Will it be?
Yeah.
Under the conditions that we use, we usually like them to be quiescent, but yes, they can be.
So they have sarcomeres.
They have sarcomeres. They're less well organized, but they do have sarcomeres, yes. So what's unique about the Purkinje cells? So obviously some of their electrophysiological properties are different. So within the heart, there are many, many cell types. There are pacemaker cells, for instance, in the SA node and in the AV node that are critical for impulse formation. There are atrial myocytes, and these are specialized for rapid activation as well as contraction. The Purkinje cells, to your point, don't really need to be. They're specialized for conducting the current and bringing it very rapidly and synchronously to the ventricular myocardium. And then the ventricular myocytes obviously need to have synchronized contraction. And then there are a number of other non-excitable cells in the heart, the fibroblast, endocardial cells, and so on and so forth. And each of these cells that have their specialized function, their action potentials in many ways are specialized for that activity. So the nodal cells have this slow, spontaneous activation. The remainder of these cells are fairly quiescent at rest. The Purkinje cells, as we'll get back to, can have intrinsic automaticity. But these have a lot of inward current, particularly the sodium current, that allows for their rapid properties to conduct throughout the myocardium. And so what underlies this? Obviously, their repertoire of ion channels, right? And so, again, in pacemaker cells, there's the pacemaker currents. They have a particular isoform of connexin so that they're not particularly well coupled. In contrast, the other excitable cells have a lot of the sodium channel, SEN5A, and the amount of the sodium channel correlates with the degree of their DVDT and the rapidity of conduction in that particular cell type. So the Purkinje cells in some ways have this somewhat confused identity in that they have high levels of SEN5A, they have a gap junction channel, connexin 40, that has a high conductance between them. But they also have the pacemaker current, and they have T-type channels that you might find in some of the pacemaker type cells. So they have a little bit of the slow and a little bit of the fast conduction properties built into that. And underlying this, these gene expression signatures are different transcription factor signatures that lead to these programs. And so the nodal cells have low levels, in some cases no levels of TBX 2.5, sort of the master regulator of cardiac myocytes, and they have certain expression of TBX5, TBX18. The others all have high or very high levels of NKX25 and different levels of some of these factors. And in some ways, it's not all or none. Depending upon the dose, for instance, of TBX5 being higher in Purkinje cells, it can set off a different gene expression program or higher levels of some of these target genes. And so we've been very interested in sort of sorting out the relationship between these transcription factors, the channels, and then their physiology. So the sort of...
Sorry, maybe you will get it to it. So you listed those sodium calcium channels. Those are depolarizing channels. Do you have a slide for potassium channels?
I don't have a slide for it, but I can share with you all the gene expression profiling we've done about differences in expression. And there are differences as well. But I've more been focusing on the depolarization and conduction velocity and fast conduction here rather than the repolarization phase. But I can give you those data if you're interested. Some of it's published.
That would be really nice.
Yes, for sure. So, the overall theme of the lab is to understand the development of the specialized conduction system, really from sort of germ cells, from the germ cells through embryonic fetal development. And if we can understand some of the pathways that are utilized here, as we progress to disease states, some of these may get re-expressed and revert sort of to these fetal forms and inform us about some of the processes that are going on. So we want to identify the gene regulatory networks that regulate conduction system development, physiology, and pathophysiology. And through this, ideally, in a perfect world, we could identify potential new anti-arrhythmic therapeutic strategies. Whether these are ion channels, whether these are receptors, or whether they are other parts of pathways. So I'm going to summarize Stacy's thesis in 2 slides, which is an enormous amount of work. But when Stacy was an MD PhD student in the laboratory, she discovered and worked on a mouse. It actually was an accidental mouse that was originally created to study the Ingrail-2 gene in mid-hindbrain development. But through sort of serendipity and an insertional mutation, it ended up being a really lovely reporter of the specialized conduction system. And Stacy characterized that gene and then went on to show, and I'll show one more slide of this, that neuregulin was very important for the specification and the development of these cells in the conduction system. And so this is sort of the key figure from the first paper, again, showing that the reporter gene was expressed in the mid-hindbrain as it was supposed to, but because of it being an insertional mutant, it was also expressed in the heart, in the specialized conduction system. And Stacy's very lovely detailing of its expression pattern over development until we got to these neonatal stages. And really the second major finding was that neuregulin was a secreted ligand that was very critical for formation and specialization and specification of the Purkinje cells. So this is an embryonic heart and the LAGZ reporter. When you cut the heart open, you can see in the trabecular, sort of the inner region of the heart, is nicely lit up. And when you add neuregulin, you can basically convert a lot of these working myocytes to assuming a Purkinje-like phenotype. And so that was 2001 and 2002. Stacy went on to greater things, and we sort of had this project on hold for quite a while, and we wanted to get back to that, and I was a little scary that it's 15 years later, but what I want to talk to you about today is mechanistically, I want to tell you now, how does neuregulin actually work? How is it regulating conduction system development and function? And so I'm going to tell you about a story that we published late last year in the JCI. And then in some newer work, I'm going to talk about how ETV1 can actually modulate cardiac conduction in different contexts. I want to tell you about a second transcription factor called EBF1 and how it's involved in specification of the ventricular conduction system. And we're not going to talk about that probably in the interest of time. So I showed you from Stacy's work that if you add neuregulin, you can get this induction of the LAGZ reporter gene. So really the first question was, okay, that's a reporter gene, but is the bona fide program, the bona fide transcriptional program for real genes actually getting activated? And so, again, these are these embryonic hearts. This is the trabecular inner layer, and this is the more compact zone on the periphery here. This is NKX25, so it's expressed in all of these cells, but it's much more robust in the trabecular zone, which is going to become the Purkinje fibers. Connexin 40 is also enriched there, and the sodium channel, again, it's in all the cells, but it's enriched there. So what happens when we give neuregulin and stain for these? You can see that just like the LAGZ reporter gene, it turns on all of these targets. So neuregulin actually seems like a bona fide inducer of this program, and it's not just sort of an artifact, really, of the LAGZ reporter gene. So next we wanted to dissect out how is neuregulin doing this. And this is kind of like those drug company charts with 6,000 different pathways in them. But we tried to simplify here. Neuregulin interacts with a heterodimer of, in the heart at least, of ERB2 and ERB4. And then there are all of these possible downstream signaling pathways. And we really wanted to identify which one of these might be active and mediating the effect of neuregulin in these cardiomyocytes. And so what we did is really go back to Stacy's assay of taking these embryonic hearts, putting them in culture, and then just adding chemical inhibitors to basically each of these kinases and seeing which of them might block the effect that we're talking about here. So pathway inhibitor, and see if we could prevent the LAGZ from turning on here. And so we have chemical inhibitors basically for each of these pathways. So we started here by adding PP2, an inhibitor to the SARC arm here, and really it didn't do anything. You can see here that we still got nice induction with neuregulin, so that kind of would rule out this pathway. Next we looked at a PI3 kinase inhibitor, and again, we didn't really see any effect there. We still got nice induction. And finally, we went down the MAP kinase pathway here, and you can see that that really blunts the induction of the LAGZ reporter gene, suggesting that we're starting to come down this arm of the pathway. So we wanted to work our way down further and sort of dissect out further downstream. So we added an ERK1/2 inhibitor. Again, it's still blocked here, so that was encouraging. And then we added this inhibitor to the RSK pathway, and again it was blunted. And finally we added an inhibitor to MSK, again blunting the activation of the LAC-Z reporter gene, suggesting that this whole pathway was being used here, converging down through activation of RSK and MSK kinases, which move into the nucleus and can serve as transcriptional regulators. So that was kind of encouraging in bringing us down sort of mechanistically to what's going on. Okay, so what are the potential downstream mediators of RSK and MSK? So this got things a little bit more complicated because there are a lot of things to choose from here, and we weren't really sure how we were going to tackle this because there aren't necessarily easy chemical inhibitors to each of these. So we tried to combine this with a sort of second parallel strategy and look at hearts and try to identify which one of those downstream factors might be enriched in the developing heart. And then compare notes with that. So basically, we used laser capture microscopy, which is a way to micro-dissect out the trabecular part of the heart or the compact zone of the heart, separate these, and then we basically could do Affymetrix chips or RNA-seq and see which transcripts are enriched into the components. This is the Conduction EGFP reporter mouse that we developed. And here we can just easily FA.C.C sort the cells into the green and non-green, and look at the Purkinje cells and the ventricular myocyte separately, and then again do Affymetrix chips or RNA-seq, and try to look at differentially expressed genes. And from that, we basically use a Venn diagram. We did this at E10.5, embryonic day 10.5, 12.5, and in postnatal day 56. And there were basically 8 genes that were enriched in either what's going to become the Purkinje cells or the bona fide Purkinje cells at all 3 of these time points. And these are all of the factors listed there. Now, if we compare that to the MSK and RSK targets that we found through the kinase inhibition assay, you'll see that there's only one factor that shows up in both of these. So this was sort of gratifying that these 2 strategies, these sort of 2 orthogonal approaches to gene discovery were leading to just a single gene. And so we became very interested, of course, in ETV1. So what is it? So it's a transcription factor. It's from the ETS family of transcription factors. And most of these, just like it looks like from our kinase inhibition assay, are downstream targets of kinase pathways, particularly the MAC kinase pathway. And so these kind of transcription factors get phosphorylated. That tends to allow them to move into the nucleus and then bind to their sites and do their thing. And Tom Jessel's lab published the knockout quite a number of years ago now. And ETV1 is actually strongly expressed in nerves. For those of you who are old enough will know that the conduction system was originally thought perhaps to be nerve-like or even neural crest-derived. And I'll show you a little bit later on. There actually are a lot of similarities between Purkinje cells and neurons. But the fact is these mice died with a neuromuscular defect, which also was sort of a nice hint that maybe we were on moving in the right direction. But there was never any role, there was really no cardiac phenotype ever described with these mice before. So we took our Conduction EGFP mice and dissociated the cells into Purkinje cells and working ventricular myocytes, and then just did RT-PCR to see if ETV1 is actually expressed there. So our positive control is the Conduction 2 gene, which is what's driving this. Obviously, it's very strongly enriched. It's about 800 to 0. I mean, it's really dividing by 0. A Connexin 40 gap junction gene, very enriched, and ETV1 completely segregates into the Purkinje cells, and there's virtually no expression in the myocytes. This is GAPDH loading control. And you can see if we take these mice and section them, the GFP nicely lights up the Purkinje cells. ETV1 is exclusively expressed in the ventricular compartment in these same Purkinje cells, and this is just the merge there, the overlap, again showing how expression was completely restricted. So we went back to basically all of these factors that were candidates and did RT-PCR to look at their patterns of expression during development from E10.5 through P56. And you can see really all of the others, they're kind of constant. There's not much change with development. But ETV1 is expressed as early as E10.5, and it just gets more and more enriched in the specialized conduction system. So again, some evidence that this was a really viable candidate. So we got hold next of ETV1-lacZ knock-in reporter mice. And in many ways, this is a phenocopy of Stacy's CCS-lacZ mice. The main difference being that there's fairly robust activity in the atria, as well as in the ventricular compartment, the trabecular cells, that are going on to become the Purkinje cells. So ETV1 is essentially reading out the fastest conduction cells in the heart. So Purkinje cells are fastest, and atria are second fastest, right? And it's sort of segregating those out from ventricular myocytes and from nodal cells. So this also became a really very useful reagent as well. So if the neuregulin ETV1 signaling axis is active, we should be able to provide some evidence of that. So this is staining kind of from the ligand through the receptors down to some of the target genes. And you can see here that neuregulin in red is in the trabecular cells, right? So it's being expressed and secreted by the endocardium, right? And it's landing on the trabecular muscle just below it. And you can see if we look at phospho-ERB4 and phospho-ERB2, meaning that the ERB receptors are getting activated, you can see again it's in the trabecular compartment and it's sparing the compact zone of the muscle. If we go downstream now, so ERK is getting activated, so phospho-ERK is there, ETV1 is exclusively expressed there, and then the target gene, such as connexin 40, is also getting transactivated there. So sort of we're going through the whole pathway there. So that, again, makes a lot of sense. So neuregulin, in theory, then, should activate ETV1 expression. And so here's a control heart, here's a neuregulin-treated heart, and here, instead of looking at the CCS-lacZ reporter, we're looking at this ETV1-lacZ reporter. And again, you can see that neuregulin is turning on expression of the ETV1 transcript. And if we do antibody staining, we can see that the protein is getting turned on as well. And so here at baseline with neuregulin, we start seeing accumulation of ETV1 in 1 day and 2 days after incubation with the neuregulin. So that also makes sense. And mechanistically, how is this working? So this is a cell that is getting exposed to neuregulin, and you can see ETV1 is mostly sitting in the cytoplasm, but when we add neuregulin to it, it starts moving into the nucleus, right? So it gets phosphorylated due to this neuregulin pathway, translocates into the nucleus where it can start actually acting like a transcription factor. So we end up with a working model of neuregulin sitting here, secreted by endocardial cells, landing on the ERB receptors, causing expression of ETV1, and then somewhat in parallel to this transcription, we also get activation of this MAP kinase pathway, this cascade of kinase events, all the way down to RSK and MSK, which phosphorylates the transcribed ETV1 and translated protein, and then can land on target genes and start activating this fast conduction pathway. Okay, so if
Do you know which are the direct targets?
we know NKX25 is, and I'll show you some of that. We also have a lot of attack-seq, RNA-seq, and mutants and differential, so there are a lot. So if ETV1 is critical, there should be a phenotype in the ETV1 knockout mice. And so we did EKG recordings in wild-type and null mice. This is an EKG in a wild-type mice, and this is an EKG in a knockout mice. And you can see the P wave is spread out, and the QRS is spread out. You actually get an RSR prime, like a right bundle branch block, and this is quantified here. So there's slowing, basically, in the fast compartments. There's atrial slowing, as indicated by the P wave broadening, and the QRS is broadening. And in about a third of the mice, they get this right bundle branch morphology. So this is very consistent with the expression profiles. And if we do intra-cardiac recordings, sort of a His electrogram, we can say the same thing here, so that we get HV prolongation, right? We get AH prolongation here, and you can see that here quantified. Again, so through the atria, and then through the specialized cardiac conduction system. So what's the basis for the slow conduction, right? And so from here, we did a lot of immunohistochemistry. So just to orient you, here's a cartoon of a heart, and most of these sections here are at the top of the septum, where you have the bundle of His bifurcating into the left and the right bundle branches. So that's where we're sitting up here. Here's the septum, here's the His, and it's bifurcating here. So this is stained with antibodies to NKX25 in red. Contactin-2 is our readout so that we know where the conduction system is. and then DAPI just to see the nuclear morphology. So in wild-type mice, NKX25, as I mentioned before, it's in basically most all of the myocytes, but it's very robustly enriched in the fast-conducting specialized cells. In the ETV1 knockout, it's really markedly reduced. There's no enrichment now compared to the working ventricular muscle. The same thing with connexin 40. We see very robustly enriched here, and it's essentially almost gone. Again, there's no connexin 40 or very little in the working muscle, and now the Purkinje system here is sort of acting the same way. And the sodium channel, again, enriched in the fastest tissue, and it kind of gets homogenized. It looks like it's all ventricular muscle now. It's not acting like specialized conduction tissue anymore. And if we go more distal into the actual Purkinje cells, again, NKX25, we can see it in the nuclei here, and it's essentially gone. Connexin 40, it's virtually gone, and sodium channel, it's reduced, right? So we've lost the key players that are involved in rapid conduction in the His-Purkinje system. So this is essentially just quantified here now at the transcript level. So NKX25 in green are the Purkinjes, the atria, and the ventricle. And essentially we have a homogenization, right? Everything is acting now like a ventricular myocyte, so kind of this intermediate level. It's obviously much more than a nodal cell, but it's nowhere near as robust as the atrial cells and the Purkinje cells we have. And we see that for NKX25, we see the same thing for the sodium channel, and we see the same thing for connexin 40. There's none of that at baseline in the ventricle. So what about the conduction system itself? You know, is there just a reduction in some of these target genes, or is there actually a problem with specification of the conduction system network? So if we take our contacting EGFP reporter gene and crossbreed it with the ETV1-lacZs and make nulls, so now homozygous for this, so that it's making no ETV1, or in this case heterozygous, right? So here we can see a very robust, complex conduction system in the ventricle here. In the ETV1 knockouts, it's very hypoplastic, right? Particularly out here in the more mid and the distal areas, we lose all of this complexity, right? So we're just dropping out a lot of these Purkinje cells. So a very hypoplastic phenotype. And if you take these knockout hearts and actually dissociate them, and in the few remaining Purkinje cells, the remaining GFP positive cells, and record sodium currents in there, just like the gene expression would suggest, we sort of get this loss of specialization. So at baseline, Purkinje cells have the most sodium current, atrial cells have the intermediate, and the ventricular myocytes have the lowest level. Basically, the Purkinje cells and the atrial cells all get reduced to the same level as ventricular myocytes. So they're not so special anymore. So does it matter? All right, so here's a mouse model. So we wanted to know if there was a human clinical correlate. So Dan Roden, who I think most of you know, I'm sure he's visited through, is at Vanderbilt, and they've set up sort of the inverse of GWAS, something that they call F.A.C.C, and essentially all the patients that come into their Vanderbilt healthcare system, they look at all of their diagnoses that are entered into the electronic medical record, and then they do a whole exome chip on all of them. And so normally when you're doing a GWAS, you know, you're kind of looking at you know, the phenotype and genotype correlation. Here they're kind of switching that on its head, and we could say, okay, if we look at this gene, and if there's a sequence variant in this gene, if there's a SNP in this gene, what diagnoses are associated with it, rather than the other way around. And so if we did that, and you looked at a SNP in the ETV1 gene, so this is kind of the equivalent of a Manhattan plot. Normally you're looking at chromosomes along the bottom and then the p-values along the side. When you do a F.A.C.C, you look at a disease along the bottom and then the probability that it's associated with that SNP. And so there's a lot of background here, but the highest, statistically most significant is bundle branch block. Bundle branch block and left bundle branch block in African Americans and other heart block in European Americans. So that was pretty unbelievable, right? When you think about the hundreds and hundreds of potential diagnoses, the number one that comes out is heart block, given what I've shown you about what's going on in the conduction system. So that was mostly in the paper. What we've done now is try to sort out if this whole phenotype is cardiomyocyte cell autonomous. ETV1 is expressed in nerves, it's expressed in other tissues. Is this all, you know, sort of a function of what's going on in myocytes? And so to do that, we took an ETV1 floxed mouse and crossed it with an alpha myosin heavy chain Cree, so we would just knock it out in cardiac myocytes. And again, we see very similar conduction slowing. We see lengthening of the P wave, and we see lengthening of the QRS, and we see the right bundle branch block in a subset of these mice, and you can just see that again in the RSR prime. So much, perhaps not all, but much of this phenotype is indeed because of ETV1 being expressed in these cardiomyocytes during development. And again, we get the same sort of hypoplastic development of the conduction system. Again, so here's a wild type, right, and the ETV1 conditional knockout, and compared to the, do I have this labeled right? Yeah. You can see much less complexity in the conduction system. So this is on the left side, and here's on the right side. And again, in the wild type compared to the knockout, you can see dropout of large areas of the ventricular conduction system. So again, reinforcing the fact that this seems to be cardiomyocyte cell autonomous. So can you kind of go the other direction? Can you induce a fast conduction phenotype if you ectopically express ETV1? So we started by doing this in neonatal rat ventricular myocytes and either infected them with a GFP control or with a virus that expressed ETV1 as well as eGFP. And here now we're just stating, so GFP is present basically in everybody. So here's connexin 40 at baseline control, and you can't see it unless the light's turned down, but there's little red speckles here that look like very nice gap junctions. So these cells are actually turning on connexin 40. This is a little bit more obvious that this is sodium channel staining, and you can see sort of a faint red blush here for the NAV 1.5, and it's really pretty markedly activated here as a consequence to forced expression of ETV1. We've also now done the same sort of experiment. So this is just quantifying the sodium currents here. So again, this is the control with just the GFP virus, and we get a doubling or so of sodium currents when we add an ETV1. This is quantified here on the right. This is sort of a display of the gene program that's altered when you overexpress ETV1. So these sort of volcano plots highlighted some of the genes that are activated when you overexpress it and some that are reduced, but ETV1 obviously is way up. Many of these are genes that are associated with fast conduction in Purkinje cells. So this was kind of reassuring. On the other hand, some of the nodal genes are going in the opposite direction. So it's really kind of toggling these myocytes between kind of a generic ventricular myocyte phenotype and pushing it toward the fast and away from the slow. Now, this is kind of an interesting plot. So this is our gene profiling of bona fide Purkinje cells versus ventricular myocytes for transcription factors, muscle and nervous system development, cell cycle, a bunch of ion channels to get back to your question, some of which are potassium channels in here, cell adhesion proteins. So, you know, these are the enriched versus the ones that are compared to the myocytes. And then compared to that is ETV1 versus the control. And you can see basically what's red in the bona fide Purkinje cells is the same thing that's red when we overexpress ETV1. So it's really giving this almost identical transcriptional signature by overexpressing this one transcription factor. So I think, again, this, you know, whether it's a master regulator, it's clearly a key transcription factor for fast conduction in the heart. So we wanted to take advantage of the fact that now we can sort of very readily separate either embryonic, fetal, adult, diseased Purkinje cells from working ventricular myocytes. And so when we went back to all of our gene profiling at different developmental stages, we wanted to see if there are any other factors that might kind of stand out as interesting. The ETV1 was sort of, how shall I say, more of a genetic screen. We didn't have any preconception what we were going to find, but with success on that, we kind of wanted to go back maybe more with a little bit of a candidate gene approach. And one of the genes that stood out was something called EBF1. EBF1 stood out because it's enriched in both the 12.5 and the P56, but also because a lot was known about EBF1 as a transcription factor that was very important for lineage specification. So it's a helix loop, helix transcription factor. And this is sort of the key factor that's important for B cell development, for lymphocytes. There's a very large and expanding literature on this, and it's been implicated more recently in other lineages like osteoclast and fat cells and even renal glomerular cells, And there's some recent data suggesting that EBF1 might act like a pioneer transcription factor. What that means, it's landing on chromatin kind of early on and opening it up so that other transcription factors can come in and remodel and cause tissue-specific transactivation. So we did some staining for EBF1, and it's got a really kind of interesting developmental pattern. So again, this is stained for connexin 40 to show you the inner trabecular layer, so that's where connexin 40 is, and it's absent in the compact zone. So EBF1 is kind of broadly expressed in the heart in both of these compartments at E13.5. At E16.5, it's kind of moving out of the compact zone and getting enriched in the trabecular zone. By postnatal day 1, it's kind of gone from everywhere except the new Purkinje cells, almost completely overlap with contactin 2. And that remains the case subsequently to all adult stages. It's really very enriched and exclusively expressed in the Purkinje cells. So we looked at EBF1 knockout mice, and from an electrophysiological point of view, in the whole animal, it had a very similar phenotype to the ETV1s. We get broadening of the PR interval, P wave, QRS, and again we get this RSR prime, right bundle-like morphology in a subset, about a third or so, quarter or a third of these mice. We've also started doing some patch clamp recordings of the Purkinje cells from these mice. So this is a wild type action potential, and we see some remodeling. We're just trying to sort out what actual genes are contributing to this, but we get a broadening of the action potential here quantified by APD 50 and 90. Resting potential is not changed at all, and the action potential amplitude is not changed at all. So we still need to sort out what the basis of this is, but there's clearly some remodeling. There's also very abnormal electrical activation in these mice. So we were talking a little bit about humans versus mice. We typically see breakthroughs, a right ventricular and a left ventricular breakthrough. Usually the right's a little bit before the left. For this audience, I don't really have to show these pretty pictures, but these are what some of the optical maps look like in a wild type. But when we look at the mutants, they're kind of all over the place, right? So this is one knockout example. The right just doesn't seem to be activated. It's all kind of starting on the left and spreading from left to right. This one seems to be getting activated way high up, and there's no left ventricle, left apical or right apical breakthrough. So they're really abnormal. And so we thought, well, let's go on. If there's this electrical activation problem, does that actually even translate into a mechanical problem? So these are echocardiograms. You guys are doing a lot of these also. So these are actually strain imaging, sort of speckle and strain imaging. What we've done here is use some software that we've got these nice kind of vectors here showing which way the strain is going over time. So you can see in a wild type mice, it's contracting and relaxing kind of synchronously. So it's a kind of a nice way to demonstrate that. And in the knockout, it's not doing that. You can see there's just this massive dyssynchrony where you've got some segments are kind of coming in while others are going out. It's just kind of willy-nilly. And it presumably is a reflection of abnormal activation in the Purkinje system. And if we actually look at this phenotype is even more dramatic than the ETV1. So again, a wild type, and here there's just these big areas of dropout. And so it's a very severe phenotype. Now, the EBF1 mice are somewhat runted, and I always get concerned when you kind of have this global phenotype, whether some of it is due to not just Purkinje cells, it also might be to some of the working myocardium. We've started trying to address that by doing conditional knockouts. The first one we've done so far is MESP1 Cre, so this is expressed in mesoderm. So it's going to knock it out in the heart, right, in all the working myocytes. It's not going to knock it out in the blood at all. It's not going to sort out yet, and we're doing other crosses, sort of the Purkinje cells from the working ventricular myocytes, but we're kind of closing in on that. And I think what you can see here, that it's certainly a mesodermal or myocyte cell autonomous property here, because the phenotype, here the right bundle is missing, right? It's still a fairly dramatic phenotype, losing a lot of the conduction system. So we'll have more to say about that when we do the rest of the crosses. Again, QRS is prolonged even in the conditional knockout with the RSR prime, and QRS duration is prolonged. So, what's sort of going on in these hearts? So we're trying to understand why we have this aberrant formation of the conduction system. So when we FACS sort, when we dissociate the cells in these hearts and FACS sort them into the Purkinje cells and the working ventricular myocytes, whoops, I don't know if I can go backwards. Okay. So about 1% of the cells are in the Purkinje fraction. If we quantify that in the EBF1 knockouts, we lose about half of the Purkinje cells. So those are somehow not getting specified, or at least they're not making the contactin GFP reporter, so we can't find them. So that's one problem. There also seems to be an abnormality in proliferation. So this is staining for Ki67 in the wild type and the EBF1 knockout. You can see there are a lot more green nuclei in the knockout. This is a reporter mouse called F.A.C.C green. There's a F.A.C.C green and a F.A.C.C red. Green means that they're proliferating, and when red is turned on, it means they've stopped proliferating. But you can see in the wild type at this stage, there's some green nuclei. There's a little bit of proliferation going on, but look at all these green nuclei. So there seems to be some imbalance between proliferation and differentiation, and cells of the specialized conduction system normally seem to withdraw from the cell cycle a bit earlier so that they can go on to become more specialized. And maybe loss of EBF1 is interfering with that process. So they're proliferating, but they don't know to sort of terminally differentiate into Purkinje cells. So there's a lot of transcriptional reprogramming. This is, we have sort of full RNA-seq, but you can see here there's changes in genes associated with heart development, NKX25, GATA4, SRF, NF2A, HOPX. All of these are down a lot, MINK is down, and some of the cell cycle regulators are up. So there's a lot of reprogramming that's going on in these cells, in these knockout hearts. And it suggests that this balance between proliferation and differentiation is getting altered because of loss of this factor. We're just starting now to do attack-seq and chip-seq and every other expensive type of seq to dig down into this. But what you can see here are these, again, neonatal rat myocytes transduced with either control virus or an EBF1 virus. This is the control attack-seq. And you can see there's a huge number of genes that are opening up when EBF1 is around by the attack-seq. There's a small number that aren't changed, and there's a couple that seem to be downregulated. But it suggests that EBF1 may, in fact, be the pioneer transcription factor getting in there and unwinding the chromosomes a lot so that other factors like ETV1 in fast conduction cells or other transcription factors in slower cells can then go on and do its thing. And so we've just started to look at that. So these factors open chromatin and remodel it by interacting with this SWI-SNF complex, this chromatin remodeling complex. One of the sort of central players there is a protein called BRG1. So EBF1 in theory could come in and BRG1's in. All of these associated factors, these BRG1 associated factors kind of come in with ATPase activity and can unwind the chromatin. So BRG1 is sort of the central figure here. I told you it interacts with all these histone modifying complexes. Interestingly, it's essential for normal B cell development, which really gets back to its role in interactions with EBF1. Knockout of BRG1 is really early embryonic lethal during the peri-implantation phase. So we've started to look, does EBF1 actually interact with BRG1 in the trabecular myocardium? So this is stained for BRG1, and you can see it's kind of enriched in this nice gradient from the trabecular cells out to the compact zone. EBF1 has the same kind of pattern here. And so if we merge them, and this is a blow up of this area, you can see that BRG1 and EBF1 seem to co-localize in the nuclei in the trabecular compartment. And if you do IP westerns, you can pull down EBF1 with BRG1 and the converse. So they seem to physically associate, be at the right place at the right time for sort of taking on this function of opening up chromatin. So we're doing one other line of investigation to kind of test the role of ETV1 and EBF1. We've started making CRISPR mutant mice where we can kind of surgically dissect out different regions. So in the NKX25 gene, which is sort of, this is the upstream region shown here, a lot of enhancers have kind of been identified here through LACZ reporter genes. There's a site way upstream here that includes 3 ETV1 binding sites and a single EBF1 binding site. And we've started making a series of mutant mice where we kind of take out 250 base pairs that include all 3 of these ETV1 sites as well as the EBF1 binding site. And this is just a PCR assay showing that these mutants are missing that, and so they're smaller by about 250 base pairs. We've made a bunch of founders. So these are these NKX25 mutant mite with the transcription factor binding site mutation. And you can see this is wild type EKG, and in the mutants, we get the same phenotype again. They get a right bundle branch block, and they get prolongation of their QRS. And if you look at some of these mice histologically, the right bundle branch just seems to be gone. So it's pretty amazing to me that you can just remove DNA from some enhancer and get this pretty dramatic phenotype in the conduction system. If you look at the Purkinje cells here, you can see that here's the contactin reporter, but the connexin 40 is gone as well. We've now started making point mutants in each of these sites, and it looks like even just removing, sort of 2 nucleotides from the EBF1 binding site is also giving QRS prolongation, but it's kind of early days for those experiments. So, here's kind of the model we're working with now. You've got closed chromatin. You've got a binding of a transcription factor, a pioneer transcription factor. It unwinds the chromatin by binding the rest of these proteins there. That leads to cell cycle arrest. And then I think context-dependent activation of different programs, specialized programs. And this can kind of go in 2 ways. I think in certain cells, if you can get actually a slow program, if TBX3 is around and sort of some transrepressors, you can get activation of a nodal program. If you're in more of the trabecular region of the heart, you can maybe get ETV1 binding there where it's active and lead to activation of the FAST program. But this is still conjecture at this point, and we're going to need to sort that out. So just to summarize, I guess this goes without saying, but I will anyway. So the ventricular conduction system is critical for normal cardiac excitation and ventricular contraction. The profiling has identified a number of novel factors, transcription factors, that are critical for the formation and the function of the conduction system, including this neuregulin ETV1 pathway, as well as the EBF1 transcriptional gene regulatory network. And loss of function mutations actually produce VCS hypoplasia. And there's aberrant electrophysiology at the cellular level and at the organ and whole animal level, really with pretty profound conduction system phenotypes. The F.A.C.C studies suggest that these variants actually are not just sort of crazy mouse models, but are actually going to have some relevance for human conduction system disease. And, in a perfect world, modulation of these sort of gain of function, where maybe these pathways are downregulated with human disease, maybe we can restore those in some sort of therapeutic approach. That still remains to be seen. There are a whole bunch of other targets I was mentioning at the beginning, other transcription factors, receptors, regulatory proteins that are enriched or exclusively expressed in the Purkinje cells that might give a handle for Purkinje-specific arrhythmias, where you could kind of, just like we have drugs that might treat ventricular arrhythmias and worsen atrial fibrillation or the other way around, you really want to have a therapeutic handle. You want to have cell type specificity. And so some of those would be really nice. I wasn't really going to talk about it, but it's a pretty picture. We're also looking at profiling microRNAs. So these are embryonic stem cells that have 2 reporters in them. They have a red one that turns on when they become myocytes, and they have that same contactin GFP reporter that turns on when they become Purkinje cells. So if you take ES cells and let them spontaneously differentiate, just like in the heart, about 1% of the cells become Purkinje-like. And you can see that by the green. When we look at microRNAs that are differentially expressed, we find some that are enriched and some that are depleted. For the ones that are underexpressed, if we expose an antagonist for that microRNA, we kind of get this sort of phenotype. So we think that microRNAs can help convert working ventricular myocytes maybe to a Purkinje phenotype, conceivably to an atrial phenotype. And microRNAs is kind of nice because they're influencing a whole system of genes rather than one at a time. Again, this is kind of early going, and we'll see how it goes, but I'll do a little show and tell. I'm going to skip these because there's just a lot of examples. It's getting late. So let me thank particularly F.A.C.C, who's a graduate student, and David Park, who's a junior faculty member, who've really been spearheading the ETV1 work. David, F.A.C.C has been doing the ETV1 and the EBF1. He's a really talented graduate student. Eugene and Karen have been involved in some of the EBF1 work and the stem cell work. Camilla